So hello everyone, my name is MyDuyen from the Zarnescu lab. Today I'll be talking to you guys about my research, which has to do with the role of aging and stress in neurodegeneration. My project and the lab that I work with has a general focus around neurodegeneration that's ranging from ALS to FTD. My team specifically, we look at aging and stress as it relates to neurodegeneration. So this type of stress we're talking about, that's osmotic pressure, electric shock, heat shock, starvation. And so we hope to find our answers by looking at stress granules in both the rat and the Drosophila, which are our model organisms. So for my presentation today, however, I'll be focusing on the rat as our model organism, and the stress is electric shock. But before we get to that, we need to talk about stress granules. What are they? RNA stress granules are non-membrane bound proteins, RNA protein granules that form when translation initiation is inhibited or during stress. So they preferentially sequester long, poorly translated RNAs and a diverse set of proteins, RNA binding proteins and others, and they vary by cell to cell. And they have been shown to be neuroprotective and increase fitness. However, if left unchecked, they have been linked with neurodegenerative diseases. So we know stress granules can be beneficial, but if left unregulated, they could cause some trouble. But what does this have to do with age? All right, guys, some bad news. We are all aging right now. And so what do we know about aging and stress granules? We know that stress granules have been seen to become more insoluble with age. As you age, your proteins get misfolded more, and that increases the number of stress granules you have. All of this, with the reduction of forming new stress granules to combat stress, contributes to neurodegeneration. This leads to my next point, neurodegeneration. Age is the biggest risk factor for neurodegeneration. So for the aging team, our project was to profile the dynamic changes in stress granules during aging by examining the expression of genes critical to stress granule dynamics, as well as translation initiation. And so we ran Western plots of brain samples taken from the cerebellum, the hippocampus, and the prefrontal cortex of rats from multiple age points. The proteins we mainly looked at were PAPP, TIAR, and G3BP1. So these proteins are essential for stress granule formations. So what does the data tell us? Data that we gathered showed that expression of key RNA granule proteins are dependent on age, brain region, and stress response. Varying levels of stress granule components could impact formation and dynamics of stress granules even in cells, even though we can't directly measure them. In aging, varying dynamics could contribute to decreased stress resiliency. And in stress, alterations in the expression of these stress granules could represent a feedback loop where long-term stress could alter future stress response. So what's next? In moving forward, our project, we will be looking at Drosophila as our new model organism. And so further investigation will isolate stress granules from Drosophila to examine how components associated with these stress granules change with aging and stress, as well as how those conditions impact stress granule dynamics. And so one of the cool things about this is we have a Methuselah Drosophila fly, which is known to have extended lifespan. So it would be interesting to see how extending one's life could change the stress granule dynamic, because we still would age the same way. The flies would still age the same way. That's it for me. Thank you guys for listening, and I'll be taking any questions if anyone has them.